I'm a little nervous that I'm talking in the first session. I was really praying that I wouldn't be, and now I'm feeling really bad about it. So I thought I'd have a few talks. I get a sense of like the spirit of the conference, and then I would adapt my talk to this. And obviously that didn't happen. And so, you know, we're going to go through it together. And I would appreciate it, you know, because I come from math, not from biology and not from physics. I'd appreciate it if you just kind of like were very performative. Like if I did something and you were like, I really don't understand the point of that matrix, make that expression. Yeah, that one. Right? So, and we'll get through this together. Okay? So how did this start? This started when someone got me sold. Our collaborator, Karen Aleem, who's been studying this organism for a while, got me sold on studying this beautiful little slime mold. Its name is Physarum Polycephalum, and it is quite exquisite. In nature, it lives on wood and under the leaf litter layer, and its lifestyle involves constantly searching, constantly foraging for new nutrient sources, and once it finds nutrient sources, linking them together to make networks. And that's where the math came in, the networks that it builds. Okay, so it's gorgeous. I love it. I love looking at it. And in fact, recently, it's kind of captured the public imagination. So there was a exhibit at the Parc Zoologique. Is that right? Okay. Merci. De Paris. On this, like, all of the weird aspects of its lifestyle, right? The networks that it builds, its peculiar kind of intelligence, right? It doesn't have a brain, it doesn't have a nervous system, but it's clearly doing something with intention, right? And how does it do that intention? How does it make the decisions that it makes? Okay? And so our collaboration kind of broke into sort of two areas which advanced in different ways, and I'm going to focus on the work that we did at UCLA, but I want to highlight the amazing stuff that came out of TUM as part of our collaboration. And they were looking at how nuclei move around within these networks. And I'll talk a little bit about the source of this propulsion of nuclei, but what you can see here are these specks that they figured out how to visualize for the very first time. Each of those specks is a nucleus. So this doesn't look like any kind of human or plant or animal cell. There are squillions of nuclei all sitting together within a common cytoplasm, right? And you can see that some of them move and some of them stay. I don't know if you can see my cursor, but there's some that just kind of end up getting stuck there. Right? And so we were looking at this from the context of how are decisions made between these nuclei. You have like a college of nuclei, right? And each of them has some level of autonomy. And you know from real life colleges that when you give people autonomy and they try and make decisions together, sometimes it goes well and sometimes it doesn't. And so how does it make these decisions? We at UCLA, working with Karen, were more interested in what the decisions were. Right? So we were looking at the networks, and here you're going to see a visualization of one of these networks. And I don't know how well you can see at the back. Can you see kind of color changing or grayscale changing at the back? I'm looking for performance. I'm looking for nod. Right? So you can see this of like the colors changing, and that reflects the fact that the tubes that make up this network are pulsing. Right? And as they pulse, they're pushing fluid through the network. As I squeeze one tube, I push fluid out from that tube, and it has to go to other places. And if I do that in the right way, I can create large-scale flows, right? And that's what was propelling the nuclei that you saw in the video before. Okay, so before I sort of talk about the behavior, I want to acknowledge all of the amazing people who worked on this project. The people who are squared, rectangled in blue here, Karen and Alex are the main people that I got to collaborate with on the calculations that I'm going to show you. Okay, and so when you sort of tackle the basic question of how do I make flow by squeezing tubes, there's two strategies that you naturally arrive at if you think about it for a while. Right? One strategy is if I just imagine a single tube, hopefully this video will play, if you look at the one on the left, if I take a single tube, I could squeeze it at one end, open it up at the other, and then I could reverse, squeeze and open, right? And if I do that, I'm going to get a flow that goes back and forth, right? That's called shuttle flow. The other possibility is I could squeeze in one place, and then like the way that you squeeze toothpaste out of a toothpaste tube, I could move where I'm squeezing along the tube, right? And that's called peristalsis. So that's a visualization on the right there. Right? So, which one is Physarum actually doing? Well, there are two camps, and they're equally adamant that they are correct and that the other camp is incorrect. So one camp believes that Physarum is doing shuttle flow, and one camp believes that it's doing peristalsis. Right? So if we look at these visualizations and we go back to our video, you could see why it'd be very hard to figure this out. Right? If I play this video again, what do you think? Peristalsis? I actually, on the weekends, I masquerade as a third grade teacher, so I bring that energy to all of my talks. So like, peristalsis, thumbs up for peristalsis? Shuttle flow? I don't know, right? You know, this is a room full of distinguished scientists, right? We don't know, right? So a lot of what we were trying to do is to figure out how we could analyze the data on what was going on in these networks to determine which was really happening, right? And so the framework of mathematics that we used for this was just to study the basic fluid dynamics of how you make flow, right? So imagine I have a tube and I squeeze at one point in the tube. So just that little section on the left here. I don't know if there's a pointer? Yeah, I'm a little shaky, but I squeeze at one section of the tube here, right? And in that little section of tube, we get what we call an extensional flow, okay? In that, there's no real pressure gradient created in that part of the tube. Okay, but I know that if I'm pushing down here and I'm squeezing out fluid, that fluid has to go somewhere. So if I look further down in my tube, I'm going to see what looks more like a unidirectional flow. And that's going to be propelled by a pressure gradient. So there's going to be a difference in pressure that pushes fluid along this part of the tube that's a long way from where I was squeezing. Okay, and I'm going to try and consistently use blue for short-range stuff, the flow that I create within my tube, and red for the long-distance flows that I create away from where I'm squeezing, right? And so at the risk of oversimplifying and bringing too much third-grade energy, we kind of think of this as being the useful flow, right? This is the flow that I create that travels long distances. This is the flow that I'm only creating locally to where I'm squeezing my tube, okay? So we can look at our whole network and all of the patterns of contraction, and we can figure out how much short-range flow there is and how much long-range flow there is, right? So my network can be modeled as a graph, right? And the graph is filled with edges, and each edge is being squeezed or opened in a certain way. And I'm also monitoring on all of the edges the amount of flow that I see in that edge, right? And this is the slide that I regret because it's early and there's a lot of matrices in it, but the matrices show up later, and so I'm still in two minds. We'll work it out as I give my talk, right? So we can calculate the relationship between the flow on each edge and the amount of squeezing that I'm doing on each edge. And there's just going to be a matrix that takes me from the edges where I'm doing my squeezing to the edges on which I'm measuring the flow. And I can calculate that matrix via a bunch of auxiliary matrices. There are rather a large number of them, but this is not amazing stuff that we did. People who've been modeling hydraulic networks forever know exactly how to do these calculations, right? So I have to figure out the pressures in my network. Remember, they showed up before, and that involves solving a mass conservation equation, and this is the most important matrix. It's the Laplacian, right? The Laplacian shows up in all kinds of areas in graph theory, and that's somehow related to this vector here of where I'm doing my squeezing. Right? And there's just a bunch of intermediate steps, but at the end, I arrive at a single matrix, and the matrix takes me from my squeezing to my flow. That's all I need. Okay. So remember I said I was interested in the long-range component of flow versus the short-range component of flow. If we want to understand the relative contributions of those things, we can calculate the amount of energy that each of them dissipates. And in fluid dynamics, we think of that as a way of just measuring their significance. So there's two types of energy that I need to think about. There's the short-range one, remember that's always in blue, and then there's the desirable long-range one that's in red. So I get two quadratic forms that I have to add together. It's just vectors multiplied into matrices into vectors again, giving us numbers, right? And then the total amount of that stuff is the amount of energy that the organism is using up, right? And we probably, I mean, the last talk was much better at this than I would be, but we think that there's probably some limit on that, right? So the sum of these things is going to be conserved. Nothing that the organism can do to change that. But it wants more of this and less of that, right? And so that's naturally measured by the ratio. And so what we asked is, what would it look like if I was trying to create as much long-range flow for a given amount of total dissipation, or equivalently a given amount of short-range flow? What I'd want to do is maximize this quotient, right? And that turns out to be a very lovely matrix math problem, the sort of thing that we do kind of routinely in math departments. All right, so we have to solve it. It's called a generalized eigenvalue problem. The important thing is there's a bunch of special ways of contracting, and those ways can be demonstrably and mathematically proved to be the optimal ways of contracting. Right? And so what do they look like? Let's try and play a video. This is a uniform network, and what we did is I'm visualizing by the color here. This is a synthetic network. It's not a real Physarum. I'm visualizing what's being squeezed and what's being expanded. Right? And those of you who've sort of taken that first university physics class may see that this looks a lot like the wave patterns you get on a drum, right? And there's a lot of mathematical similarity between the equations that describe the wave patterns on the drum and the equations that describe the flows on this network, right? And there's a certain spectrum. We can measure the efficiency of these modes against the number of the mode, and there's a certain power law, and in math we love power laws, and all of this stuff can be shown, right? So now we apply it to the real network. Okay. So what are real slime molds doing? Right. So it starts by looking at the videos, and just to sort of show you, that we really can see these contractions. What you were seeing before is just kind of a change in intensity. Hopefully it becomes a lot clearer when you zoom in on one of these videos. You can see the tubes opening, tubes expanding, tubes contracting, right? And all of that is done in an oscillatory way. And so what we do is we take our real network and we calculate the optimal modes for that network, and we project the real contractions and expansions of the tubes onto those optimal modes, and we try and figure out how many modes the real network is using. Okay. The answer turns out to be about 4, right? So this is the data that you saw before, now visualized using modes, not looking at the whole video, but looking at just the specific modes that we saw, and there's a little video that shows the modes changing over time. Oops, doesn't want to play. Right. This is how much they contribute to the total dissipation in the network. This is the number of the modes, and you can see that it's really only this is a log scale, right? So these are really not very important at all. It's only like these modes, right? We're not very good at counting, right? What's that, 4, 6, something like that, right? It's not a huge number of modes that the network is actually using, and we can visualize them. Again, it looks a little bit more complicated than the network that I showed before, which was totally homogeneous, but they're fairly simple. They correspond to things moving left to right, right to left, diagonally, maybe in the other diagonal direction, but we can project everything down onto this simple basis. So this was our first victory. Like, rather than having a whole video showing all kinds of complicated looking things, we can just characterize what our network does in terms of a few modes. How important are those modes at this moment in time, right? And we can see how the modes change when the network gets fed, right? So this is the network being fed, and you can see a lot of change in the shape of the network. Hopefully also you can see some sort of change in the way that it's pulsing. And we can visualize that if we measure the amount of energy in each of our initial modes. At the moment of feeding, there starts to be a change. We're initially mode 3 is the most important mode, but mode 1 gets promoted. So rather than having to think about all the network and all of the complexity, all we have to think about is the interplay of a few modes. I hid this at the bottom because I don't like wading into scientific controversies. That's like my least. It's not. I love it really. But I don't really want to be I don't know, it's been a difficult time at UCLA, you might have noticed, so I don't want people yelling at me, but we did not find any peristalsis. So those of you who put your thumbs up and said it was shuttle flow, we agree with you, right? That's as much as I'm going to say. I'm not going to say you were right, but we got the same thing as you saw. And those of you who saw peristalsis, you can go write your counter paper, and we can just keep this this train rolling for as long as we like, right? So we can visualize all of the flows in the network, and we can start to use this as a basis for understanding how much behavior the network has, how many modes it's using at any one time, right? And so when we got started on this, we were thinking, like the goal of this, and I forget exactly the language we put into the proposal, was like the genius of Physarum, right? How intelligent it is. And like the one way to think about this is, it's pretty amazing that of all of the strategies it's located, the ones that it's using, the modes that it's really using to push flow through the network are optimal, right? So that's a lovely way to think about it. Is it hard to find an optimum, though, right? Is it a challenge? So, you know, we lie awake a little bit worrying about this, and we've kind of convinced ourselves that maybe it's not, right? So this is the unfortunate coda to this talk, and the unfortunate coda to like letting mathematicians look at your problems. And so this is, what we did is we asked ourselves, could this be, you know, an emergent behavior? Could this be something that the organism just self-organizes to do? And so what we do is we go back to our graph model here, right? And now we're going to think about all of the ways that the tubes could contract, right? So I have a tube or expand. So this is the volume of a certain edge of my network, and it's going to contract, this particular one. And it's going to sort of change as a function of time. Okay, and, okay, I know it's too many equations, I apologize, right? And so just ignore the right-hand side for a second and just focus on this thing, right? So we know that it oscillates over time. The volume of each tube oscillates over time, and the equation for an oscillation is just this little thing, right? So you have, you know, this would be a harmonic oscillator or spring oscillation. But we know, so what this would do would just have the tube passively oscillating in volume over time. What we expect is that there's some feedback between the pressure in the tube and its volume. And so if we incorporate this feedback, and I grayed something out, I had enough sense to gray out a little bit of the math, but if we incorporate this feedback from the pressure on the volume, we can show that there's some energy. I'm really regretting all of this, right? We can show that there's some energy, right? And that energy is going to grow over time, right? And that's because of some particular sign choices we made. We decided that when the pressure was low, you would keep expanding, and when the pressure was high, you would keep shrinking, right? You're really pushing into like bad pressure. And if you do that, then the fastest growing modes, the most unstable modes that it tends to select for are the ones, and this is why I felt like I needed to show the matrices, and then I regret that. These are precisely the eigenmodes of this Laplacian that we introduced before. So it's very straightforward for it, so long as it follows. I realize it's masked a little bit by the math, but it's a very simple rule for using pressure feedback to change how quickly your tube radius changes over time. So long as you use that feedback, naturally you locate these optimal modes. Okay, so that's my last result. So what did we find? We found that Physarum contraction patterns are optimal for internal transport, which is fantastic. Is this optimal contraction patterning a sign of an indication of intelligence? Maybe not, I say in this safe space. Maybe it's a fairly easy thing to self-organize. Although, you know, as a mathematician, just because I can write down an equation doesn't mean that the biology to, you know, realize that model wouldn't be exquisitely complicated. A couple of things that continue to keep us awake at night. How does the slime mold produce any kind of pumping that isn't optimal, given that it naturally makes like the most optimal pumping? And so that's not too hard to answer. And then the next thing, can we quantify the network formation behaviors? So could we go back to that original video, showing it foraging, showing it pulsing, showing it continuously building new networks and modifying and adapting those networks over time? I would love to be able to say the answer to that question is yes. But this is really like our open frontier right now on this problem. So thank you.
So thank you very much for an entertaining talk on a really fundamental problem here. And we have time for some questions, please. Yes.
Yeah, thanks for the great talk. I was wondering, you mentioned that you didn't see any evidence for peristalsis, but how would the eigenvector, the modes for peristalsis look like?
Yes, thank you. Thank you so much for asking. I don't need to stand that close, do I? Yeah, sorry. So, yes, I'm so sorry. It's been difficult at UCLA. So, I didn't show the data for this, but like you can make, so let's look at our modes, right? Like you could have a mode that does this, or you could have a mode that does this, right? They both look like standing waves, and if I just used them individually, all I could do was produce a shuttling. If I put them together with the right phase difference, then I can make a traveling wave, right? So in order to look for peristalsis, what we need to do is not look just at the amplitudes of our modes, but look at their phase relationships. And so our evidence, I'm not going to say evidence against peristalsis, or rather our failure to find consistent peristalsis is that the modes tend to be very lockstepped in phase. They tend to come on and off with exactly the same phases. Without a phase difference, you can't get any kind of peristalsis.
Okay, please.
Yeah, fantastic talk. Continuing on about the peristalsis versus the other possibility. I guess the network topology is changing all the time in this organism.
Yes.
Right? And the data you showed, it looks like you're just working with more or less a fixed topology. So I'm wondering how much it depends on the network topology, the answer to that question.
So there's a couple of answers to that question. Like one of the things is, the answers is yes, and one of the answers is no. So it's sort of a little messy. Everything is messy. So I should have known this when I got started in biology. But the yes answer that topology matters is sort of clearly the overall shape of the network is changing, right? And clearly the distribution of mass in the network, you could see when we fed it, or when Karen fed it, like it started to move mass in a really significant way to the right. And so like its shape really, really clearly is sort of influenced by the pumping in the network, right? And like once it gets into foraging, you know, the topology is changing all the time and you can't see a single cycle, our theory is currently useless. And I guess I apologize for that to HFSP. But the other answer is no, and the no answer comes from the fact that like if you take the network and you rearrange all of the tubes, but you keep the overall shape, or you change the hierarchy in the network, none of that stuff really strongly affects or really influences in any significant way the spectrum of optimal modes in the network, which was a big surprise to us, and it's linked to the fact that like under the skin of this is an operator, the Laplacian, that doesn't really care very much about hierarchy. And this was a sort of big pushback, I think, to a community of people, including myself, who thought that hierarchical networks were important and interesting because they were hierarchical. That's not really significant here. And so like to that extent, like if you take the network and you keep the overall shape the same, you know, and you change the topology within that shape, how things are connected and the particular wiring pattern, doesn't really have a huge influence, at least empirically and, you know, within our ability to study the effect of that on the Laplacian. So that's my no answer.
Okay, thanks.
Okay, we'll take one last question, please.
Thank you very much. It was interesting. The question that I had is, regardless of the transport mechanism, you've got feeding on one end of the network, but there has to be things that are very far away from the source of the feeding, and there needs to be some mechanism to basically trigger that action. Is that understood?
So this is kind of linked to my how does it produce, that's a much better way of framing. I wish I'd had you look at my slides. There's a much better way of framing this second from last point that I wanted to make, which is, you know, like there's a certain set of optimal modes, but sometimes you don't want to use those exact optimal modes. You want to move all of the food to the right, or all of the mass to the right. And it turns out, like we haven't, 70% of an answer to that question. It turns out that it's what you do at the periphery of the network. So if you start to soften like the periphery of the network, you create these things that the people who study slime molds call fans or lamellipodia. You can change the optimal modes, right? And so that's part of the self-organization, and it's not something that we had time to talk about. But the short, pithy answer, and I know I've gone long past pithy, answer to your question is that you can make a local decision near where you're feeding the network, which is just soften here, and that will naturally create a global response. So it's really beautiful in that respect.
Okay, so
thank you.
I guess we should probably use a coffee break for further interactions and questions, which I see in the audience. But before that, please join me in thanking all the speakers for some very interesting talks.